Hello everyone, my name is Nabanita Chattopadhayay and I am serving as an assistant professor in the Department of Biotechnology, Haldia Institute of Technology. Today in our genetics lab class, we will be preparing different stages of mitosis from actively growing root tips of onion or Allium cepa. Mitosis is a kind of nuclear division or karyokinesis where a mother cell produces two identical daughter cells actually having exactly same number of chromosomes like the mother cell. For performing this experiment, we need actively growing root tips from onion. For getting those type of actively growing root tips, first we have made a few scratches in the region from where roots can grow. Then we have placed those two onions over two conical flasks filled with water. It should be noted that the water must touch the scrapped region. After 3 days, we have got actively growing roots from those two onions. From each growing root, we have cut about 1 to 2 centimeters from each root and we have taken those root tips in a water-filled watch glass. For performing the experiment, we need a few things like blade, forceps, scissors, watch glass, slides and cover slips, carmine stain, acetocarmine actually, hydrochloric acid, carnoise fixative solution, etc. The cut root tips were taken from the watch glass and were placed inside the carnoise fixative solution and kept in 4 degrees centigrade for overnight. On the very next morning, we have started our experiment in our laboratory. First, we have kept everything ready for our experiment and cleaned the glass slides and watch glasses properly. Meanwhile, we have taken the root tips out of refrigerator and kept in one Falcon tube and placed that in the rack. The alcohol present in the fixative solution will stop cell division and will fix the cells properly and will dehydrate those. For the experiment, we needed one normal HCl. So first we have prepared that. With the help of a brush, we have taken out the root tips from the fixative solution and placed them in one normal HCl. A little stain residue was present in the brush, so the hydrochloric acid solution has become a little pinkish in color, but that won't create any problem in the experiment. The hydrochloric acid will help form pores in the cells so that in the next step, the acetocarmine stain can penetrate the cells and can reach chromosomes. To make this more effective, we have passed the watch glass with the root tips and HCl over the flame of a burner and then we have kept that for about 5 minutes. For the next step, in a fresh watch glass, we have taken 500 microliter of acetocarmine stain. Then we have taken those root tips out of HCl and placed them inside acetocarmine stain. For more effectiveness, again we have moved that acetocarmine stain with the root tips over the naked flame for 5 seconds and then kept that for around 15 minutes. After 15 minutes, we have washed the root tips twice. In a watch glass, we have taken clean water and then placed those root tips in clean water. After washing for 2 times, we have started our experiment for preparing the slide to watch different stages of mitosis. About 1 drop of acetic acid is taken on a clean glass slide, and then one root tip is aligned properly on it. We can clearly see a small part stained very dark in that root that actually will be taken for our experiment. The deeply stained part will be taken and the rest will be discarded by the help of a forceps and a blade. When a clean cover slip has been placed on it, it should be noted that no bubble should be there inside the cover slip. Then this whole set will be taken inside a blotting paper and will be tapped gently by the help of fingertips, and a smear has been formed. As we can see clearly, a smear has been formed. Now another student is performing the experiment. Now we will check this region for mitosis. First, we have seen that in 100 times magnification so that we can set the field, and then we have seen the cells in 400 times magnification. Interphase, prophase, then metaphase, anaphase, and finally telophase. We have seen all the stages in different slides. Thank you for watching.